Greeting dear judges, my name is Chandra and I'm here with my friend
Gabriella Isabelle.
We're from SMP Santa Laurentia and today we will present our research entitled The Efficacy of Bintaro Leaf Extract for Cerberin as a bioinsecticide for weaver ants or Oecophylla smaragdina.
Have you ever heard of weaver ants? Weaver ants or Oecophylla smaragdina are ant species that are often found in tall trees with fruits. When the fruit from the trees wants to be harvested, sometimes they feel disturbed, so they end up biting and injecting formic acid, causing itching and irritation of the human skin. Therefore, an insecticide innovation is needed to eradicate this ant species, which is more eco-friendly than for its surrounding. In making this bioinsecticide, use bintaro leaves or Cerberin as a raw material. There are several reasons for the use of this plant species. One of them is that this tree is very abundant in nature, where the status given by the IUCN Red List is least concern. In addition, this species has no economic value, so it will be in vain if it not used properly. Lastly, the Cerberin leaf contains substances called secondary metabolites, such as terpenoids, flavonoids, and iridoids, which are toxic and repellent to insects.
This research is a quantitative experimental research that uses the process control group design approach. For Cerberin leaves, we take them from a tree in the Santa Laurentia school area. The leaves were washed with running water until clean, cut into small pieces and dried in the oven for 12 hours at a temperature of 40 degrees Celsius. After that, the leaves were ground using a blender and dissolved in 70% ethanol at a ratio of 1:10 for 24 hours in a region bottle, or other words, the maceration method. Then, the solution was filtered using a glass funnel lined with filter paper until the extract was obtained. Cerberin leaf extract is stored in the refrigerator until the time of use. The leaf extract was dissolved in distilled water at concentrations of 0%, 25%, 50%, and 100%, where the solution with a concentration of 0% only contained 70% ethanol. Then, the solution was sprayed on the Oecophylla smaragdina sample using a hand sprayer in a petri dish, and the mortality was recorded after 10 minutes. Analysis and determination of LC50 are done through SPSS 25.0 software and probit analysis. We had done phytochemical screenings for saponin and also flavonoid. After the experiment, the mortality of Oecophylla smaragdina was found at different concentrations of leaf extract. At 0% extract concentration, there were no deaths in ants. Then, the 25% extract concentration showed a mortality of 28.57%. Extract concentrations of 50% and 100% killed 66.67% and 83.33% of the ant samples, respectively.
Terpenoids are natural defense substances that are generally toxins, feeding deterrents, or oviposition deterrents. This deterrent interaction is triggered by the emission of volatile terpenes, which is included in volatile organic compounds, or VOC. The emission of this substance is a way for plants to communicate with their environment, one of which is to issue a deterrent signal and become an elicitor to stimulate the defense mechanism against insects. The next secondary metabolites are flavonoids, one of example of which is rutin. When rutin is swallowed by the insect, the glycosidic bond will be hydrolyzed and release quercetin. Quercetin inhibits the ATP synthesis in cell mitochondria and inhibits the cytochrome P43 mixed function oxidases. This will cause a deterrent effect and growth inhibition on the insect body. Iridoids, the latter secondary metabolite, are substances found in 50 different plant families. As a plant defense agent, this substance can be activated by beta-glucosidase and become a very strong protein with a protein cross-linking and lysine decreasing activities to reduce the nutritional value of the insect food. And it will be a feeding deterrent.
So it can be concluded that the best concentration of Cerberin leaf extract as a bioinsecticide of Oecophylla smaragdina ants is 100%, with the highest mortality of 83.33%. In addition, the LC50 of the extract was also obtained, which was 38.6%. From the conclusions obtained, we suggest to conduct the extraction of Cerberin, a cardiac glycosid, which can be done using chromatography.
And here is the references we use for our research.
So that's all we have to say. We apologize profusely if there are any mispronunciations or things that offend. Thank you for your attention and time. Have a nice day.